Thank you very much, Gayatri, for that introduction. Um, it is really a distinct honor and really a pleasure to be speaking in this lecture series. Um, I think I might be probably the only one in the lecture series who does not have a photograph with Professor M.S. Swaminathan to show how close I was. um, but suffice to say that my association, well, I can't call it association, my acquaintance, personal acquaintance with him does go back quite a long time. We met in, I think, 1996 for the first time. Um, and that time I was at the John Innes Center in England. And then later, of course, we met in a couple of conferences and then a couple of times while I was at IRRI for nearly 17 years. He came over twice, and so we met there. And I then had the rare opportunity to be able to actually write a small article in the ICAR memoirs for Professor Swaminathan. And I think it's what I wanted to really highlight is the human aspect, because what I could see during his visits, all the people that thronged to come and say hello to him and give him a hug, and he knew the names of all the kids, so let alone the staff, he knew the names of their kids. So I was totally blown off because of that. Um, and it was Dr. Kush actually who introduced me to Professor Swaminathan in England, and I think that humanity and humane aspect of him was something that really stuck with me. And I did come here last year, I think, or maybe a little while before that, with Dr. Umesh Singh, and he was not very well. I could not see him, and I really regret that. But I think what is important is that, as this slide shows, that he had this amazing capacity to really look at the work that is done and be able to extrapolate its importance. And I think those are the kind of lessons we all need to take as scientists. We cannot just be happy with what results we get from our studies, but what is the importance, and what are the potential applications, etc., and how they can change life. I think that was uh, really central to his visions, and that's how he could make such a big difference. Um, and one way of saying that is that, you could extend this quote, which is a very common quote: we are not only what we eat, we are also how we grow it. And that kind of extrapolation is extremely important, because how we grow rice, and especially rice, because it has so much effect on climate, etc., and also it's the crop of so many of the poor people. So how we cultivate, how the environment is affected, how the personal health of the farmers and the customers is affected, is really at the core of this whole issue. And again, rice typically provides a number of examples for that. And going ahead with that, the drought tolerance in rice has provided, at least for my understanding, a lot of insights into how things might work. And I apologize in advance that this seminar might be a little more technical, but don't worry about trying to understand the slides. There is a message in each slide, which I hope I will be able to bring out. Um, and again, wrap it up at the end to be able to say what all this really means. Um, but coming back to this drought issue, I think if you call it the worst hydrometeorological hazard, it wouldn't be wrong. And you can see on your right-hand side, in the last century, nearly it has killed 11 million people and adversely affected 2 billion people. There are a number of direct and indirect impacts of that. And of course, we are familiar with most of those. But to us as scientists trying to look at drought tolerance, what is very interesting is that over the years, people who have been looking at drought tolerance have come up with genes and proteins that are involved in many different pathways. And for a very long time there was this idea that if we find that one big master regulator which then controls other genes, then we might be able to have drought tolerant rice or drought tolerant crops. But what we have ended up with really and that is what I would like to demonstrate here today in a little more technical way, but the message remains the same, that it's a really complex trait. While most of the traits would involve so many different genes, there's hardly a trait that you would get a master regulator for. Drought in itself becomes extremely complex. And so I've listed all the different kinds of protein categories and gene categories that people have come up with. What you also see a little above that, the papers in last 35 years. So, I did this exercise about 2 or 3 years back, nearly 3 years back. And if you put these words, drought and plant with an asterisk in the Web of Science, you come up with a certain number of papers. Okay. And then you then kind of home in on, drought and improvement in the title. And you get about 600 papers, and nearly 105 of those papers actually in the title say that they have found a drought tolerance gene. Okay. And yet we don't really have that gene-based drought tolerance till date in any crop, right? So what that tells us really is that we have to go further ahead from this, single gene processes. And what is the way of doing that? So how do we really have a fresh look at a dry problem, as this slide shows? A scientist called Gary Atlin, who some of you might have heard of, either as a scientist or most likely as the BMGF person who has been funding a lot of rice work and crops work, actually, especially about creating new varieties from the Gates Foundation. He was at IRRI early 2000s, and he came up with this idea that we should not worry about trying to get the plant to survive, but we should worry about whether or not it can yield anything under drought. So if a 6-ton, 7-ton per hectare line can give you 1 or 1.5 tons, that's still something. And only once we have those kind of plants, then we can take our understanding of the molecular biology and the breeding aspects further ahead. And with that, there was a lot of success over the years. Of course, all these QTLs that are listed, quantitative trait loci, these are genomic regions, large genomic regions. And when I say large, it can be anywhere from 1 centimorgan to 8 or 10 centimorgans, actually. And so there are a number of genes in those areas. But they found number of different QTLs. And the way these QTLs are listed, you could see QDTY 1.1. So DTY stands for yield under drought. Okay. And 1.1 means chromosome 1 and the first QTL. And 2.1 means chromosome 2, the first QTL. So then as you go down on chromosome 2, you have another one. Again, you have another one, 2.3 and chromosome 2. So similarly, you keep going down. The first one in this line was DTY 12.1. And if you look at the literature, there's a lot of information on 12.1, the breeders, how they have used it. And my group started working when I went to IRRI in 2008. We started working on trying to figure out the molecular aspects of this QTL. So I will be essentially describing to you how we went about trying to understand the molecular aspects and the functional aspects of this QTL. But then as we established that understanding, what became clear with time also is that it's not just the genes and their sequences, but there are structural aspects to how these QTLs work. And so I'll mention a little bit about that. And this is where I came up with the title of trying to interface the functional genomics understanding of genes and genetic regions and the structural aspects. Most of it is about the functional aspects, because what we have been able to do is something unique, which is that um, there is a lot of understanding, or at least now, there was not when we started looking at this QTL. Now there is an understanding that there could be multiple genes involved, but nobody had actually shown what those genes are, what they do, and really try to validate them. And so most of the talk will be actually a quick effort to show you that we understand some of those genes, not all, nowhere close to all, but just the fact that we can show you 6 to 7 genes and their functionality and what they do really brings home the point that multiple genes, even within the same QTL, are extremely important. And we started doing that when I went over there. There were not the facilities and the people were not there. I had a very small group of 3 people. And so we did a lot of bioinformatics analysis. And without going into the details of what this schematic diagram is all about, what it really shows you is that all along this QTL, the small peaks you see are the peaks of different genes. Okay, so we go through that genomic region, we understand what genes are there, we try and see if there's any literature on those genes there and whether the literature relates it to drought tolerance in any way. But then there are other bioinformatics aspects. We can look at the SNPs between the donor parent and the recipient parent. We can look at the genetic and protein interaction networks, and we can look at the cis regulatory sites and protein domains of those proteins, because these are all predicted, and there are softwares that you can access to ask these questions. And depending on how rich they are, either in their networks to drought-related genes, or do they have in their promoters cis regulatory elements, or do they have in their proteins specific domains which have been already shown to be drought tolerant, we gave them a certain scoring. And based on that scoring, when you come up with a weighted average, you get these peaks. And so in this case, we get a peak on something called the OS-AH, and AH stands for amido hydrolase. But the more interesting part of it is in the lower figure, which is that because we knew the SNPs, we could actually see which of these genes are polymorphic between the donor parent and the recipient parent, which means that they can now serve as markers. So earlier on the top line of the bottom diagram, you see there are only 5 markers that were used to get this QTL. And then there were more SSR markers added, which is the second line, the yellow line, which has the yellow part. But then we were able to add all these gene-based polymorphic markers. And these came out to be extremely useful for us. Do you want me to go up there? No. Okay. I think it's okay. Probably clear from here. So yeah, these gene-based markers came out to be extremely useful to make our point about the importance of these genes. But because we had these markers now, almost like the SSR markers, so we could sort of reconduct the QTL analysis, and through that we then come up with additional peaks quite above the lot score of 2.3, which is important. And you see again OS-AH coming up, which is amido hydrolase. There's another gene called the MTN3, which stands for a gene that is similar to another plant, which is Medicago truncatula. And at that time, it was not named any other way. Then there is a transcription factor called OS-NAM, which stands for no apical meristem. And then there is another transcription factor, the ARF. So these are four peaks at least, which are above the Lord score and may be important. But the top one is the transcription factor or SNAM. Now, NAC transcription factors in the literature are known to confer drought tolerance, but this NAM was very interesting. It is part of a group of 8 NAM-NAC factors in one of the class, and that class is defined by what is known as a pest domain. There's a protein domain called the pest domain, and there are very few transcription factors which have 2 pest domains. Many transcription factors would have a pest domain, which is shown by a green block in the very top diagram, schematic diagram, which shows DD, which is dimerization domain. Then there is a classic NAM domain. Then there is a DNA binding domain, DBD, and the P refers to the pest domain. Now, this pest domain is responsible for how stable a protein is. And if you have more pest domains, that means it is not very stable, sorry. What that means is the turnover rate of this protein is really, really high. But importantly, there is a very clear difference in the sequence of the donor and the recipient parent. The donor is the YRRM, which is denoted by WR. Recipient parent is Vandana. And you see that the difference in that sequence manifests in the difference in the protein structure also. The small white arrow in the protein structure in the 2 black diagrams, you see the white arrow showing a difference in the structure as well. So that's about the protein. But if you go to the genic promoter region, there are clear differences in the cis regulatory elements also. And most of these cis regulatory elements are associated with drought tolerance. So clearly, our analysis, bioinformatic analysis that highlighted NAM had really reasons to highlight this NAM as a very useful gene. And we tried to go ahead with that and show, its role. Interestingly, that bioinformatic analysis also told us that there are a number of genes in this QTL which are target genes for this transcription factor. So this transcription factor has to upregulate or downregulate certain genes, and it might do so, for genes spread all over the chromosomes. But even within this QTL, there are some genes which are targets for this transcription factor. So if this gets induced, it will go up and either induce or reduce the expression of these target genes. And these target genes are shown by the small circles here. That is the transcription factor. So the squares are the transcription factor. The circles are genes for different traits. And you can see that network of this one single transcription factor, how it affects many other genes. The green ones are all within the QTL. Some are directly influenced by it, and that is why they are in this beige circle, and others are outside the beige circle. And there are ways of actually testing that out. If we say that they are involved, how can we do that? There's something called the electrophoretic mobility shift assay, EMSAs. And we've done that EMSAs for all the genes that we think, for the genes that we could clearly see and putatively think that, they are target genes. I'm sure there are more which we have missed at this stage. Interestingly, all this study of EMSAs, these beautiful EMSAs were done by somebody from the foundation here who came over to IRRI for, I think it was 6 months or 9 months probably, Govindan Ganesan. And I think he's still in Chennai working at the SRM Institute. And he did a fantastic job. These are not easy to do. These are very, very difficult experiments, and he did a fantastic job. But what that really shows is that again, the bioinformatic analysis that we did and thought that this NAM should be influencing other genes within the QTL, it does so. So the classic way of proceeding ahead with that is you do a transgenic plant with the NAM transcription factor and ask the questions, what is the phenotype of these plants now? Let me also quickly say over here that we had a near isogenic line also from the breeders. And this is the QTL transferred in the recipient parent. This is called the 481B over here. So what should happen is that 481B is a line that is really clean in its genomic background, but has only that QTL region taken from the donor parent. Okay. And so it will have certain changes in its phenotype. And so when we transfer the NAM gene, we again see that number of those phenotypes are actually recapitulated. So if you look at these, the panicles, for example, which is a little more clear than the roots, what you see is YRRM has really big panicle and a lot of spikelets. Vandana does not have so many spikelets and it is not so much branched. And again, 481B is almost looking like the YRRM, number of branches and number of spikelets. You then, if you go to the transgenic plants, which is the bottom figure of the panicles, which is IR64 wild type and IR64 transgenic, you can again see that because of this one single gene, there is more branching and there is more spikelets. And similarly, if you look at the IR64 transgenic and wild type in the roots, which is in the test tubes below, you see that roots have much more profusion. So with one single gene, we could actually recapitulate some of the phenotype that we get from the entire QTL. And before I go to the next bit, let me just quickly show that the roots we could easily quantify. And when we look at these roots in the NIL 481B, which is the Vandana genetic background with the QTL from YRRM, the Vandana is the wild type, the YRRM is the wild type, but also we generated the transgenic line in the Vandana itself. So we transformed, the previous slide was showing you transgenic plants of IR64 transformed with NAM. This slide is actually showing you Vandana transformed with NAM. So that, it becomes even more easy to compare what is the role of this one single gene in the background where the QTL had otherwise gone. Okay. And what you clearly see is that under well-watered conditions, the roots are all very similar. They're all, the length, the depth, and the surface area taken together in this 3D diagram. The roots are very similar. They're all congregated together. But when you expose those same plants to the drought stress, which is DS, you see how they really separate out. And the ones that are really going out are the Vandana transgenic NAM plants, the green, long green spikes that you have. Again, proving that this one gene was very useful in affecting the root architecture. Before I go ahead, what was missing is that we did not get enough yield. The fertility was not good. So that is again the point that just this one gene is not enough. So there were some improvements in the panicle structure, in the root architecture, but there was not enough yield. The fertility was really low, which means that we are missing still something. And we went through all the major omics analysis of 481B, Vandana, YRRM, NAM transgenics. And this is one of the transcriptome analysis, which I'm trying to show you here. What came out, again, without trying to describe to you the entire picture over here, because that's thousands of genes here. What was very clear from this analysis is that within the QTL, you can get epistasis. So genes that are in one side of the QTL and other side of the QTL, they interact. And you can get epistasis within the QTL. Now, this kind of thing is known, gene from one chromosome being epistatic to gene in the other chromosome. But this was an interesting one. It was not unique. It has been seen before, but it was interesting. And again, an evidence that there are multiple genes involved. Okay. And if you then go to the same kind of a transcriptome analysis for the roots per se, you could again very clearly see some of the differences. And in this one, there is will I be heard if I go over there? Or thank you. So I just want to point out this one particular line over here. And when you get data like this, it's not very clear unless it is really, really, a stark difference between the green and the yellow and the red. Over here, you can see there are all different shades, so it's not very easy. But in some cases, it is very easy to see the expression going down from or going up from green and yellow to the red. And one of these genes is what we call the OS-WAC. OS obviously stands for Oryza sativa, and WAC stands for wall-associated kinases, right? And again, it is in the literature, it is known to be effective in drought tolerance. And there's very clear difference between the nil and the recipient parent. There's a very clear difference between the transgenic and the wild type. So this was a very useful set of data. We published all of this. We also went on to do the proteomic analysis. And again, what is very clear is that there is a big difference in protein turnover in different tissues. So each of these 3 sets of bars are for the spikelets, for the roots, and for the panicles. And you can see that under drought, so this is essentially a capture of what is going on under drought. And you see in roots, some of the sets of proteins are really upregulated in the roots under drought, but in some cases they really go down. So the red bar really goes down. And so there's this entire mixture of things going on in terms of which category of proteins will be up or down regulated. But what this basically tells us is that even with just this one QTL, different tissues are even differently affected. So just because you have this QTL does not mean that the leaf, root, and panicle will be similarly affected. So this omics, of course, it gives us a lot more details even at the single protein level. But the overall broad picture is that this QTL affects different tissues differentially. And that again is one would know that by common sense, but it's good to see it through the omics analysis. And similarly, with the metabolomics analysis that we did with that. And here again, you can very clearly see, if you look at the first part on the right-hand side, on your right-hand side, there is WW, which is well watered. There is drought stress, which is shown by DS. Now, the 3 small bars are for proline, which are hardly there, which means that under well-watered conditions, proline is not accumulated. Now, proline is a drought marker, okay? And as soon as these plants see drought, the proline levels really go up in VIRRM and in Vandana. But the red bar, which is down there, means that very little proline accumulation happens, which means the 481B is really drought tolerant, and it is showing it even at the metabolite level. And similarly for the starch and sugars. Sugars are again important because of the osmoregulations. And you can see again that the 481B under drought conditions has more starch and more sugars. So this is all about what we could do in combination with the near isogenic line 481b or with the transgenic plants of Vandana or IR64. And we were able to make a point that NAM is important, but it's not doing everything. So we move on to a second gene, and I will go ahead rather quickly with most of the other ones. And this work is also published. Now, this gene actually controls the epigenome, and it's a big network of a diagram that we have over here. Well, I hope you can see the red cross right at the bottom, which shows guanine going from guanine being transformed into xanthine. The red cross means that the gene that does that, or the enzyme that does that, the gene for that enzyme has been silenced. Okay, and so you would not have guanine being converted into xanthine. So this gene is the typical amido hydrolase. So you remember NAM had one peak and AH had another peak. So we then asked the question, what does AH do? And we had the mutant lines for that. But this guanine is actually upstream in the pathway connected to S-adenosyl homocysteine, which is the second last block at the bottom on your right, S-adenosyl homocysteine. So if guanine accumulation happens, you might expect that the S-adenosyl homocysteine accumulation also happens. Okay. Now, SAH is a very potent inhibitor of DNA methyltransferases. And if that happens, then its effect on DNA methyltransferases happens. And what that means is that the genes are differentially methylated. And we could show that through 4 different ways of looking at the genomic methylation. So we know that there is a gene involved in epigenetics as well. There's a gene 3, which has a very long name, glycerophosphoryl diester phosphodiesterase. So we always used to get confused between, is it GPDP or GDPD? We had a lot of confusion about that. But anyway, one of the PhD students called Wang Roena Oane, in the Philippines, they have this very typical thing that you never get to know the real name of the person. They have these nicknames and everybody just knows the nicknames. So Wang, we used to call her, but her name is Roena Oane. She did her PhD on this particular gene. And she's somebody, you give her a task and she can just sit there for hours and hours. And you can guess that by looking at this image that she teased out the stigma and the style or the gynoecium of many, many different plants, hundreds of different plants. And we show some of them over here. So she teased out all these stigmas. On the top, there is a schematic which shows the different components, the anther, the filament, stigma, ovule, and the lodicule. And she dissected out enough of these, stigma, ovule, and lodicules to actually do qRT-PCR. And the qRT-PCR, as shown by the blue bars, clearly shows that there is some expression in the lodicule, and there is more expression in the stigma and a little in the ovule. And before that, of course, she looked at the different developmental stages of the panicle. When does it express in the different developmental stages of the panicle? And it's the PA5 stage in which it expresses the most. And when you then gather spikelets from that stage, then within the gynoecium, it expresses most in the stigma. Now, this is a very tiresome work, but she loved it, and she had this fantastic amount of data. What was not very clear in the diagram is actually the difference between Vandana VIRRM and 481b in terms of the morphology of the stigma and the papilla, the angle between the two parts of the stigma. And again, she did that through image analysis. And again, it was very clear that the Vandana, which has the QTL integrest, which means 481b, starts looking like YRRM, which is the donor parent. And Vandana by itself, under both well-watered and drought stress conditions, is not a very strong, well, we don't know that, at least not at this stage. It doesn't look really as healthy. So YRRM stigma really looks, flourishing. So does the 481B under well-watered conditions. But under drought conditions, obviously it affects the stigma, but not as much as Vandana. And the 481B again comes up and it is flourishing. So this gene is really concerned with overexpression, with the health and the morphology of the gynoecium. Now, what does that do? Obviously, it affects the fertility, because wider the stigma, more easy to catch the pollen and more easy the pollination, etc. And so then we ask the question, what is the pollen count on these stigmas and what is the final fertility? And again, you can very clearly see that under drought stress, the fertility goes down. Under well-watered, it is quite good. But this is under overexpression of this gene, again in IR64. And so what you can then see is that the transgenic lines compared to the wild type IR64 are showing much better fertility and much better pollen count. We come to gene 4. This work was done in the lab of Professor Benildo de los Reyes, who is a good friend from my earlier times at IRRI. He used to be the technician of Dr. Brar in the next door lab, and then he went to the US to do his PhD and stayed on there and really flourished and became a chair at one of the Texas universities. I think it's Texas A&M. And so we had a collaboration with him, and he did a fantastic job of looking at another gene, which is called decussate. And this is, again, a transcription factor. And the network is actually connecting a number of genes shown in red. What is amazing is that all these red genes are actually concerned with flowering. So, you have gene for flowering, you have gene for fertility, and we were able to really clearly show that all these genes need to be there because they are affecting different aspects of what we see in our drought-tolerant plants. The next is the STPK, which stands for serine threonine protein kinase. Now, some of you might know there are a number of serine threonine protein kinases. There's, I can't remember the number per se, but there are many, many kinases, and then within them there is a class of serine threonine protein kinases. So what is special about this? This, we could show, is actually involved in the alternative splicing. So this goes and phosphorylates the SR proteins, and those SR proteins are then useful for alternative splicing. So again, going back to the processes involved in drought tolerance, alternative splicing is again one of those things which is invoked. And STPK was also different amongst the two parents, and we could show very clearly its expression patterns, etc., in both in all three: flag, leaf, panicle, and root, etc. The next one is a nitrogen content regulation gene. And what is very interesting in this case is, again, without going into any kind of description here, those of you who are interested would probably be able to have a look at what's going on. But the bottom line in this case is that there is a substrate X, and an enzyme acts on this substrate. It converts it into product 1. The reason I have not spelled out the substrates and products is because we are trying to get an intellectual property rights on this or a function of this gene. But what is interesting is that when you silence the enzyme A, which converts substrate X into product 1, you would expect less of product 1 and accumulation of substrate X, because it is not being consumed now. Okay, and we see less of product 1, but we do not see an accumulation of substrate X. And the hypothesis there is that substrate X is going through an alternative pathway and coming back to being converted into product 1 and product 3. Now, these product 1, product 2, and product 3 are very small molecule nitrogenous compounds that can move around in the plants. We have transgenic lines, we have genome edited lines of these plants, and we have been able to show that the plants have a lot of nitrogen. These plants can grow in 30 to 40% of the nitrogen that is otherwise applied in the pots, in the hydroponics, and in the field. Okay, and essentially we have generated plants that do not need enough nitrogen application. We do not need enough all the fertilizers that is added to the rice plant. Okay, so why am I not so excited about this whole thing? The reason I'm not excited is this situation is not very different from when the farmers put a lot of nitrogen in their fields. And when you keep adding nitrogen to a field going from a lower amount to a higher amount, the yield keeps increasing. But after a certain time, excess nitrogen reduces yield. And this is what is happening. We now have so much nitrogen in the plant that it is reducing yield. So essentially, the partitioning of the nitrogen to go into the grain is not happening very effectively. Actually, that's not correct. The partitioning is still happening, but because the nitrogen content has changed and physiologically there is a set ratio of carbon to nitrogen that must exist in the plant. So now we have too much nitrogen, but not enough carbon. Okay, and so the pathways that are important to take the assimilate of starch to the grains is not happening. So the yield is compromised. Okay, about 15 to 20% yield is compromised. But the good thing is we now have something in our hands to ask the question, can something bring the yield back? Okay, if we can show that, then what we are saying is that farmers can grow rice with 50 to 60% less nitrogen application, which has amazing consequences in terms of climate change, in terms of income, in terms of soil health, etc. There's the 7th gene for which we have very little information. We were able to show very clearly that its expression is changed under drought. We have not done much work on that, but another group from within IRRI used this gene. It's called the MTN3, which is also called the sweet gene. And this is extremely useful in blight tolerance. And they did genome editing to actually show that they can have multiple resistance to blight tolerance, blight strains. So that is at the single gene level. There's this one last one I want to show you quickly. We got some mutants in some of these genes, and all of these genes apparently affect the root architecture, except the amido-hydrolase. And the reason for that is that the amido-hydrolase is an overexpression line. The rest of them are silencing lines. So which means that the amido-hydrolase is a negative regulator of root architecture. So what final evidence can I show you? Apart from saying that all these single genes do different things which are important for drought tolerance, what evidence can we have at a bigger picture level that many genes within this QTL are important? And this, I believe, in my 30 years of research, is the best schematic diagram I could ever generate. So if you remember, we were able to have markers, gene-based polymorphic markers along the QTL. So what you see here is different recombinant lines. So the QTL has broken up at many different places. So each line, the QTL is broken up at 1 or 2 places, okay, or maybe more. So what you see is the dark blue is a allele for a particular gene. The genes and the markers are shown right at the top. If it is light blue, that means this gene or this allele is coming from the donor parent. If it is yellow, it is coming from the recipient parent. If it is gray, it is a heterozygous plant. And if it is white, that means we could not get data for that. But what you clearly see is under well-watered conditions, as you go down, which is the increase in yield, there is no real pattern to this whole blue, yellow thing. But as soon as you put the same lines under drought, you clearly see that although the yield is drastically reduced, but as it is increasing, the one that has the maximum yield is actually the one which has all the alleles from the QTL. It's fantastic. It's like, somebody has painted this thing. So that was one of the, I wouldn't call it the final nail in the coffin, but it was very gratifying that it was not just, us trying to convince everybody through single gene characterization, but at the breeding level, at the lines level, you could see that effect. So what have we been able to see? That there are at least 7 genes to more or less extent that we have characterized to show that they have a role in this functionality of this QTL. We have shown that in different genetic backgrounds in IR64, in Vandana, and also in Arabidopsis. The work with decosate transcription factor, they were able to show that in Arabidopsis also, Benny's group. We use transgenic plants or the nils, and transgenic plants were single gene plants. The nils were the entire QTL, so we could make those comparisons. But what is interesting now is, again, you go back to the processes and all these multiple processes. There's a hub transcription factor, which was the NAM transcription factor. There's epigenetics, there's alternative splicing, there's flowering, fertility, nitrogen use, sugar transporters, epistasis, and also post-translational protein modification. I haven't shown you the results, but the NAM transcription factor is actually sumoylated. And the reason it is sumoylated is, again, due to those pest domains. So it undergoes very rapid sumoylation and ubiquitination because of those pest domains. We have data for that, but I thought it just getting too much into the details. So there is post-translational modification also responsible for that. So is this really a unique thing that we have discovered, or are other QTLs also doing something similar? Gosh, I've taken too long. Maybe another 5-7 minutes. Is that okay? Yeah. Okay. Interestingly, another group actually has looked at other QTLs, and this is QTL 1.1, QTL 3.1, and 12.1. And they used miRNAs. And again, without getting into the detail, what this diagram actually shows you here, that the miRNAs that they got had target genes in these QTLs. Okay, and those target genes are listed for 1.1, 3.1, and 12.1. The green genes are the ones that are bioinformatically predicted to be important for drought tolerance. The purple ones have actually been tested at the expression level. So when you expose plants to drought and you look for expression, they are up or down regulated. And the brown ones are the ones actually which are common to both the approaches of figuring this out. Okay, and so 1.1 has a lot of them. 12.1 has very few, and 1.1 has very few. So one could say that, again, these multiple genes are involved because you got it at the miRNA level, which means another omics level is bearing this hypothesis out. But if you don't see many of these in 12.1, what does it say? Well, all it says very simply is that the mechanisms of the regulation of 12.1 are not within the miRNA regulatory domain, and they are more in a different domain, and I'll come to that. So apart from those QTLs itself, are there any other examples of people having discovered that QTLs could be made up of multiple functional genes? Yes, there are. I've listed only 3 over here. There are at least 7 or 8 more, but none of them have actually done, well, actually the last one, which is one of the later ones, the maize work, but our work preceded that. Nobody had shown till that time the characterization of those genes and their roles in the QTL. There were bioinformatic predictions, which is the first one, and then there were other subfractionation of QTLs kind of work, which is the second paper. And the third paper is using CRISPR-Cas methods to actually make the changes in the genes and be able to show that different genes are responsible. And so essentially what it is saying is that where we started out from with a hypothesis, not only did we prove it, but the literature is proving it now. So what are the structural aspects of this? This complex diagram, what it is showing you is that genes, when you try and track them in different genotypes, or in cultivated and in wild rices, this diagram is taken from another paper. They keep moving either within the same chromosome, they go to different areas, or they could even go from one chromosome to another. And this is a very big aspect of what we call the structural variants. These structural variants could be the DNA sequence itself or the epigenome. So because when you have epigenetics, you can end up changing the cytosine to thymine. There can be deletions within the sequence. There can be duplications, insertions. So you could have these things going on only in one single gene, or you could have these going on in full gene blocks. Okay. There could be different copy numbers, and then these complex arrangements, which is shown by this diagram over here. Now, a broad definition of a haplotype is, does a gene from one genotype look similar to the same gene from a different genotype? Okay, and you could look into these kind of deletions, the SNP markers, the insertions, inversions, etc. The broader definition of haplotype would actually also include copy number and complex rearrangements, but these are the ones that we generally go for. Why is this important? So we did some work very long time back in transgenic plants, and we were able to show something which there is a lot of literature out there, but it's not very well appreciated. I did 2 years of work in Seattle working on human genomes, and we were looking at beta thalassemia genes basically. And there are 4 genes that would express prenatal and 4 genes that would express postnatal. And what decides this gene switching has been a very topic of a great research. And nearly 20 years back or so, this concept of transcription factories came into being. Now, you all know what a nucellus is. Nucellus is an area where all the rRNA genes come to be transcribed when the transcription of these rRNA genes needs to happen. So whichever chromosome they are on, whichever chromosome territory they are on, they will loop out, and they will loop out in a manner similar to what is shown right at the corner, right-hand corner. So if you extend this, so something looking like a wire, you extend it, all these 4 black kind of parts, they are on the same length, but they will loop out to come together into a transcription factory. So the classic model of the fact that the transcription complex moves on the DNA while transcribing may not be right. It's actually the DNA that moves on a stationary transcription factory. And nucellus is the biggest transcription factory. But what is now being realized is that other genes also work through transcription factories. Why am I saying this to you? And the reason I'm saying this to you is, remember, NAM has other target genes within the QTL. And so these would loop out together, because when drought happens, all of these need to be expressed. So NAM would become part of a transcription factory, and all these genes will loop out to make a difference over there. And so basically what we are saying, that's a very, very complex issue. It's not just about the genes, it's not just about the alleles, it's just not just about the haplotypes, but how they reorganize themselves in 3D. And that 3D reorganization depends again on what is otherwise called junk DNA. We think it is junk DNA, but it may not be junk, because if you delete that, then that looping will be affected, and you will not be able to get the expression of that gene. Okay, so we really, really need everything taken care of. And something called the haplotype-based breeding strategy is something that can take care of that, simply because the haplotype means that it will ask the question whether it's the same sequence, whether it has no deletions, inversions, etc.
and whether it's the same sequence within the gene, and then are those same genes involved as a gene block? Are the genes in the gene block the same allele? All of that is taken care of. So it's one of the easier ways of doing it. And we again went back to ask the question, can we do that? And so this diagram very quickly shows you that we analyzed 450 different accessions, and we were able to get one particular haplotype, which is shown in the blue over here, which has a block of genes which are extremely important for yield under drought. And we can now use this to really introgress into other plants. If you combine that with speed breeding, which has come to a point of actually having nearly 4.5 to 5 generations within 1 year, then you really reduce the breeding cycle from 8 years to nearly 2.5 years. So the reason for that is that we are really not wanting to spend any more time even on the QTLs. Okay, so forget the genes, forget the QTLs, but if we go through the haplotype-based breeding, we are actually looking at trait efficiency loci, so TELs. So the haplotype breeding strategy will give us trait efficient loci rather than just structural aspects. This is my last slide, second last slide, before I come to the people who have been very helpful in doing this work. What this shows you is if we really want climate smart and climate resilient plants, what all feeds into it. And the biggest block over there, as you see, is of the gene bank. What we are not using completely is the biodiversity. We really need to have a look at the different aspects of which different genotypes are available for which kind of traits. And the gene bank is an extremely useful asset. And then using that gene bank, taking the genotypes from there for breeding and agronomy, and then bringing in all your other aspects of metabolic engineering, synthetic biology, the more regular ones about the breeding, the more useful ones for the microbiome and metagenomics, etc. The phenotyping is included. So, and all of this generates a lot of data. And we are now in an area or in an age where AI and ML will help us take care of this extensive amounts of data. And so unless we try and integrate these different aspects, we will have something missing. And thanks to AI, or efficient use of AI, we are now in a position to do something about it. Did I miss one? Yeah, so yeah, I talked about TEL and QTL, but the thing on the side is something that I am very passionate about. I wish I was better at maths and computers, because my second life after retirement would have been actually devoted to quantitative genetics. We have so many bioinformatics people, we have a lot of computational biologists, but we lack people who are very good at analyzing data, but who are also very good at interpreting what is the biological significance of the data that we have. And I seriously think that our education system needs to do something about generating more proficient computational biologists and quantitative geneticists. These are all the people, it's my last slide, who have been instrumental over the years in helping us get all the data that I have shown you. There's a lot more tons of data that we have on this. The PDF line is important, and all 4 of the people in the PDF did a lot of work. The PhD people, again, going from your right, Min, Dhananjay, Jenny, all of these people contributed a lot. And there were people who are visiting as either visiting research fellows or on-job trainees, etc., and who really contributed to this. Sorry, I think I've taken too long, but I hope I've been able to make a point about how critical it is under this climate change aspect to be able to understand what are the best methods and approaches to generating resilient plants. Thank you very much again.
So thank you very much for your comprehensive overview from QTL to gene and back to QTL again, why the overview is very important. So if there are any questions first, maybe one or two quickly from the audience, and then we can move on online. Anybody here has questions? So maybe I can just ask, why do you think a QTL like this has evolved and is present in something like Vandana?
Again, this is just hypothesis. I have no idea. There's no real answer I have to your question. But my guess is that something like this is not easily observable in plants like wheat and maize. Their intervening DNA there's a lot of intervening DNA. Okay, there are a lot of transposons, especially in maize, for example. And these modular structures are probably present in smaller genomes. Okay, and they have evolved because you're exposing. Vandana is a landrace, for example, and it is more easily exposed to drought than cultivated varieties. And so they have evolved in a modular sense to make sure that there is an efficient use of resources to be able to respond quickly. Okay, but let me just say, in case there are people online who know a lot more about this QTL. This QTL is even more confounding because it actually came from the susceptible variety. Yeah, it came from YRRM, which is actually a susceptible variety. So I don't want to get into the details of that. But clearly, there could be regions. And one of the things I would really like to do, if we have a good bioinformatics person, is to ask the question, how many regions are there where you have a transcription factor and you have genes which are targets for that transcription factor? And it's very easy to do through bioinformatics, except that you want to do it for n number of regions. And you could actually come up with a hypothesis there to say that rice has more modular structure than perhaps other, and maybe the wild rice, the cultivated rice has more modular structure. So for example, this bit that you see the same number of genes are in a much smaller region at the top and the bottom, which are the cultivated varieties. Okay, the wild rices have much more areas. So the cultivated rice has much more modular structure than the extra DNA that exists.
Any question?
Thank you, sir. Thank you for your insightful talk. Actually, out of 7 important genes that you have mentioned, I think the ARF one, I think
you're right,
not explored much.
Yeah.
Is it an auxin responsive factor or the one which involves in like the polar auxin transport, adenosyl ribosylation factor?
Yeah,
So
you're absolutely right. I think literature is full of those, ARFs. Unfortunately, we could not get the right person to start on that. We got taken by other things because, when you're working on a gene and it turns out to be affecting only the stigma, you get excited about, similarly, you get this nitrogen thing happening, you get excited about that. But that ARF thing has always been on our minds. Unfortunately, we could not look much into it. But I want to point out, do you see a red line on this diagram? There's a red line. Yeah?
Is this the one?
Yeah, that's the one. You see this red line?
Yes.
Yeah. This is a gram domain protein. Okay. Now, we don't know much about this gram domain protein, and there's not much literature on that. But there are 3 meta-analyses on drought responsive genes. And all of these meta-analyses put gram domain gene, specifically this gram domain of 12.1, right at the top. And so we discovered this later on after that meta-analysis. All 3 meta-analyses were published. And we would really want to go back to this gram domain gene to ask the question, what is its role? So there are even bigger and better genes than ARFs that could be very important. Could this be a master regulator? We don't know.
Thank you.
Yes. Good morning. I'm Prabhavati. So your last slide spoke about the role of microbiology and metagenomics and microbiome, its importance, but it has been very less studied about its function on the expression of genes. What are your views on that? And my second question is about the stress ethylene, which is produced during drought or salt tolerance. Is it connected to the QTLs or stress ethylene levels?
Yeah.
Can you just highlight on that?
Yeah. So for the first question, I want to keep it really short. I have my views and opinions, but this is a very open seminar, so I'll be very restricted in my opinion about that. I think it's a fantastic development, this whole microbiome thing. And the more we sort of hear about it, especially from the human side, about how important gut microbiome etc. is, we get more and more convinced about the soil microbiome also being very important. But let's not forget this soil microbiome or the plant microbiome is an interaction between two very dynamic biological entities. And so whatever solutions we will find will have limited geographical and locational applications. So I think the question to be asked is how much of our investment, time, money, energy do we want to make into this to say that any interesting results that we get, how can we make it applicable to different kinds of soils, to different kinds of crops? They are very specific. All these microbiomes are very, very specific. You spend a lot of time to figure out, and you get a lot of data. And to make sense of that data itself is very difficult. So you just go for something that is present here but not present here, and get excited about that, which is not very different from what used to happen with microarrays and transcriptomes. Like, I found this one gene, but you have data on 30,000 genes, and you're looking at only 3 genes out of 30,000 genes. So similarly, we need a lot more development of tools, technologies, approaches, models, AIs to make sense of all the data that we get through this microbiome and metagenome analysis. Yeah. The second part was the salinity pit. I don't know about salinity, but I know submergence and heat. There are QTLs which again are seeming like they would not be single or 2 gene QTL, but they are multi-gene QTLs. So I think it's a regular phenomena now, at least with the abiotic stress, that if you find some useful genomic region, it would have multiple genes over there, at least in rice. Yeah.
So would multiple QTLs also interact?
Very interesting question. And Arvind Kumar's group spent a lot of time, Shalabh Dikshit, Swami, they did a lot of work in pyramiding different QTLs. And what they discovered is that some QTLs would have additive effects, but there would be 1 or 2 which can actually pull the performance down. So the QTLs, again, the haplotype. So if you extend that haplotype at the genome level now, which is all about genomic selection, okay? So you could then go into the genomic selection aspect and then ask the question, which QTLs would be useful together.
So I think we'll move on to the online. Are there any more questions? Yeah, there's one from Dr. Swami Palani. I am from Papua New Guinea. Very nice of you, sir, to join. How can functional genomics help understand the physiological responses of PNG, that is Papua New Guinea traditional rice land races, if collected, compared to imported varieties under drought stress? Because this country is facing a lot of paddy cultivation and they importing from foreign countries. That is why he has raised this question.
Yeah. So a quick answer to that is you've really asked a very important question in terms of the physiology of the plants. So if you step back from molecular biology and genes and genetics and functional genomics, etc., what is really happening is that the physiology of the plant is changing. As a molecular biologist, you can go in and say, this is what this particular gene does. But that gene does not do it just by itself. It does it with many other genes. The proteome is completely changed. The metabolome is completely changed. Okay. So when we say that those omics changes have happened, what are we actually saying? What we are really saying is that the physiology has changed. Okay. And it's a pity that a number of physiologists have gone into molecular physiology, and there is a real dearth of field physiologists. So when a molecular biologist wants to go to a field physiologist, there should be a very close collaboration to understand what's going on. So again, the nitrogen work, for example. So we've generated something. Now we really need physiologists and agronomists to tell us what can bring back the yield in this plant, which already has something which we have sought for the last 60, 70 years, right? Interestingly, all the land races, this gene is silenced. That is why they can grow in less nitrogen. Okay. Going back to the Papua New Guinea thing, I don't know, how aware you are, but my impression over there, I visited that place. It's a very political minefield also. The rice in Papua New Guinea is a rather political issue with the Taiwanese, with the Thai, and with the Australians. So I'd rather not say anything more right now.
Thank you. Yeah, just a minute.
My question. So my question is there any other QTLs which confers drought tolerance? If so, why you have not concentrated on those?
Is there other what?
QTLs which confers drought tolerance.
Yeah, well, we can't deal with one enough. So there's a list of all those QTLs that I began with. There are other people who have looked at other QTLs. In fact, this the work on miRNAs is by totally different group, but they have worked on the same QTLs. They're not even from IRRI, actually. So these QTLs are extremely popular, both for introgression for drought tolerance and for molecular research, because their bottom line is they work. They work. And so molecular biologists are very excited about the fact that we've got something here working. How can we show what it is that makes it work? Okay. But for us, just 12.1 is, rattles our brains. So, yeah, there are other people who have been doing other QTLs also.
Sir, actually, thank you for the wonderful session. And basically, I could get an overview of the session because I'm from a food tech background. But then the public perspective, how these kind of research can be translated into the field, and what are the challenges that we have in terms of regulatory or the IP, and how the traditional seed knowledge system that across the world that we have, which are tolerant to drought or any flood perspectives.
Yeah.
Do we also include those kind of knowledge also into these kind of studies?
Yeah, very good question. Very, very good question. So if a focused answer is required, then the focused answer is some of these QTLs have been introgressed into varieties, into lines which have become varieties now. Okay. The CRRI in Cuttack has actually generated lines. I think it's called CR80 something Dhan, which has used these QTLs to make it drought tolerant. What is also interesting is that they've gone back to some very drought tolerant lines and asked the question, does this line have this QTL? Without introgressing it, just the question, would this QTL be working naturally, automatically? And yes, there are. So Shabagi Dhan, for example, has 12.1 sitting exactly there and doing that. So that's the focused answer. But I think what you're asking is a much broader question. Fortunately, the QTL introgression work does not involve much regulatory aspects. There are aspects of making sure that what you say is working well is really, really working well. And that's where the national systems come in place for multi-locational trials. And so we have this ICRIP RICE agency, which will take your new line and test it in different parts of India to be able to say whether what you're saying that this new line is good, is it really good or not? So they have that system is in place, and it's not necessarily a regulatory process, but it's a process that makes sure that not every new line that a breeder makes becomes a variety. So it's a varietal release process there, right? Now, going back to the traditional things. So as I said, with this one single gene, we have a very clear indication, the nitrogen efficiency gene. which is silenced in most of the traditional rices. Okay, so effectively, we have, when we make edits in IR64, which is a very popular elite modern cultivar, with one single gene silencing, we can almost convert IR64 into a landrace, because this edited rice has big biomass. The biomass is rich in protein. It has big roots. Okay, it does better photosynthesis. It requires less nitrogen. So these are all characteristics of land races, traditional rices. Okay, because these traditional rices are grown in less, fertilizer, less water. So they are drought tolerant. They don't need so much fertilizer. And with this one single gene, we have been able to show that. So imagine what else exists in these traditional races. But the question is, they are not very good in yield. Now, do we keep concentrating on yield, or we say that we as a country, even as a globe, there is enough rice to eat, right? So should we really start concentrating on nutritional aspects of these rices, of these traditional lines? Okay, and that's where the discussion needs to happen. Who does that? To what extent? Is there a mechanism to promote that? Is there a mechanism to take things to the market? Those are very different questions. Probably you are a socioeconomist. No. Food technologist, yeah. So they're full of nutrition. So for example, again, one of the results that we have with this nitrogen use efficiency gene, it yields less, but the grains have more protein, which is again very similar to the traditional land races. So we've converted IR64 into almost a traditional race, land race, okay, with one single gene. Now, if we can only bring back the yield, so now our thrust is from the opposite direction. Earlier, we didn't have enough nitrogen. Now we have enough nitrogen, and we have enough physiological understanding of all the photosynthesis and assimilate, partitioning, etc. Can we use that physiology and agronomy knowledge? So for example, when you have too much nitrogen, wouldn't you need to correct the phosphorus and potassium also? So one of the first experiments to do would be, let's increase the amount of potassium and phosphorus in this, okay? And then it would go back to the socioeconomics again. The saving that you're making from saving nitrogen, are you losing that by adding more nitrogen, potassium and phosphorus? That's for the socioeconomists and agronomists to figure out. So short answer, yes, traditional varieties are good. They are more nutritious and they should be used. But how do we use them? To what extent? Which areas? What kind of farmers? Are there mechanisms to make sure that farmers benefit from it? But the biggest question is, will you eat it? Because these rices have been there for eons, but we are used to eating polished, heavy starch rice. I told my sister, I was sitting there and I said, red rice, brown rice is good for you. So we brought some brown rice, we had it once, and it's been 1 month since we haven't had it again. So the biggest question is, how do you change eating habits? That's the biggest change you want, even before you actually do all this, because otherwise it will be a waste.
Prajesh, are you online? Prajesh, you had raised your hand.
Yes. I hope I'm audible.
Yes.
Yeah. Good morning. Yeah. Thank you, sir, for this lecture. So I'm Prajesh Parameswaran, a botanist associated with MSSRF. My research interests are in biodiversity governance, especially the access and benefit sharing mechanism under the international treaties and national act with respect to rice land races. So I have a very quick question. You have mentioned many donors for drought tolerance. Is there a way to trace the donor land races for drought resistance across the globe?
Oh,
Or if I put the question in a different way, I have a particular variety which is traditionally known for its drought tolerance. The variety is there in the collection of NBPGR and also in the IRRI gene bank.
Right.
Is there any possible way to trace if it has been used up for breeding new varieties, either by public or private entities in the country or outside the country? Thank you.
So I'm not sure if I'm giving the right answer here, but if you say that it is present, it is there in the IRGC collection in the IRRI gene bank, then let's not use the word trade. Simple way of getting it is just write to the gene bank manager, and the gene bank manager of IRRI is actually obliged to send you 20 grams of that. So if you want that, you already have it, but I don't know what you mean by trade. But if anybody wants that and wants to conduct research and use it in their breeding program, all they have to do is write to the gene bank manager, and the gene bank manager and his team will send you 20 grams of that, and you can then test it, whether it's the right thing. You can test it for drought, and if it works, you can then use it for your research for, being able to breed a drought tolerant variety which is otherwise popular, so that you get the trait of drought tolerance from the land race, but you get all the elite characteristics from your elite rice. I don't know if I've answered your question, but that's all I can say.
Yeah, just to make a clarification, I don't need any sample, but I just wanted to know how many varieties, how many land races have gone out for breeding.
Oh.
Just to know the implications of access and benefit sharing mechanism, how it works.
Yeah. I don't think I have the right answer, but a broader answer that I can give you is we did a few years back, we did an analysis of how many of the gene bank accessions have been used in breeding programs. And it was only 5% of the 132,000 genotypes that we have. So 132,000 accessions we have, only 5% of those have actually been used. And in many cases, they are land races. Many cases, they are land races. I mean, most of the traits have come from land races. The submergence tolerance has come from land race. The drought tolerance has come from land race. Many biotic stress tolerances have come from land races, some even from the wild rices. So again, I don't know if I've answered your question, but there are very few accessions that have been used. And that is one of the reasons why I put that block in my last slide of using the gene bank diversity. There's amazing diversity with plants that have amazing characteristics and traits, which can be extremely useful. And yesterday, during my discussions here also, one of the things I was really interested in, so over the last few months since I retired from IRRI, I've been actually going to different parts of India. And everywhere I sit and talk, they say, oh, we have this particular rice, which is a traditional rice. It's extremely good. It's very popular. And I've never heard of those things. Never heard of those things. It seems like as soon as you leave the cities, people in small districts and villages, they have their own rice. And so who is eating all the IR64 and who is eating all the MTU 1010? I don't know. But there is no catalog, at least not to my knowledge. I'm sure there is information somewhere if I get really serious about digging that out. But you end up listening about all these traditional varieties which are nutritive, which are aromatic. And when I was growing up, there is only one aromatic rice, that was basmati. But now you go anywhere and there's an aromatic rice there. So somebody really needs to have an overview and list all these things and try and, what's the right English word? Disentangle them from their colloquial names into whether or not these exist as official entries in any gene banks, etc. Sorry, I just went on a rant over there, but I think the bottom line is that very few traditional lines have been used, and it would be better if we can use more.
Yeah, Thank you, sir. Thank you.
So last question, Gopika. Yeah. Thanks.
So thank you for the talk. That was very insightful. I have a thought regarding the discussion we had about using microbiology for stress tolerance. So, as you said, it's very diverse. The microbial environment is very diverse in different environments. So, if we target the root exudation pathways, will it be a one-stop solution for the diverse stressful environments? So, can we target those?
I'm not sure. I'm not sure. When people have looked at root exudates, I don't think they have done very extensive analysis of the exudates. Most of them pick up only on some important and interesting organic acids that they have. Malic acid is one. I don't know if oxalic acid is exuded or not, but malic acid surely is something that people look at very quickly. I think the literature is scant even on, a full metabolic analysis of what all exudates are available or what all does rice exude. Okay, and it's not very difficult to guess that depending on the exudate, you will have the microbiome modified with that. And then each genotype can have a difference in exudations. Okay, so it's a very complex area. And now if you start asking the question about genotype to exudate to microbiome, it keeps getting more and more and more difficult. But again, I say the beauty is now we have AI to be able to deal with this complex data. So at least, is there any data, or do we want to generate that complex data? Because the analysis now is easier, right?
Well, thank you very much, Professor Kohli. I think it was fascinating talk covering a very wide range of important issues, and you can see from all the questions that this discussion could have gone on for a lot longer. But it raises a number of important issues and questions very relevant for MSSRF's work. Because on the one hand, we're looking at land races, traditional varieties, farmers' rights, how to ensure seed sovereignty, how do they get the credit, and so on. But on the other hand, you also have to show that these are distinct, that they have the value, that and for that, all these modern scientific techniques that you mentioned are very important. And really, I think today it's the science has evolved to the point where that one single gene or one single factor that makes a difference is not going to be possible. Doesn't exist in nature. It's very complex. And so collecting good high quality data and then using it to do good analysis, maybe with the use of AI. So partnering with people who have advanced capacities for bioinformatics, for those kinds of cross multiple data sets analysis, I think is going to be very important for the future. And you're also asked a good question about what is a choice that the country is going to make. So we've been chasing yields for a long time. Now, I think there is a realization that we have to look at nutrition, not only yield, because as you said, there is enough quantity being produced. But the fear always is, are we going to lose out? Are we going to get short of food again? And that, I think, is stopping us from making the kind of policy decisions we need to make. So I believe that this is the right time for like-minded scientific organizations to come together and prepare a roadmap for where does Indian agriculture, where should it be going? Keeping in mind rice and wheat are our staples. We're going to continue to eat them. But how do the pathway should be to build more nutrition security, where reforms have to start in agriculture, but also in the big social sector, programs like PDS or school meals, where the quality of what is being given to people has to be significantly improved from nutritional content perspective. Yeah. So I think this could be just the beginning of a dialogue. I know that many of you have also had some time with you yesterday and this afternoon as well to continue with these discussions, but it's a real honor and privilege to have you here to deliver one of the MSS Centenary Lectures. We'll be having one more in the month of December to round out the series, right, on the 5th of December at 5 p.m. And it's going to be delivered by Dr. Mary Angela, who won the World Food Prize this year for her work on soil microbiology. So that's again going to be very interesting and very complimentary. I think many questions came. So mark your dates on the 5th of December at 5 p.m. India time. So that will be 8:30 a.m. for Mary Angela. She'll deliver it online. So again, many thanks, Dr. Kohli, not just for delivering the lecture, but also spending some time with us. And we hope that we can keep building on this relationship. Thank you. Thank you very much.
So thank you everybody here in the audience as well as online. We'll be logging off now. Thank you for your participation. Yeah. So Dr. Kohli, there's just one last question. Since you've come from a long way off, I thought we'd just take this also.
Just one question, sir. Some farmers believe no, variety rice have a very short life itself. Like we cannot use it long time for food and all like that. So the farmers believe that variety rice has a very short lifetime. So how it's true or what is its assumption?
Farmers. I really like to meet those farmers because the entire research agenda is pushed by varietal replacement. You know, our funding agencies and our funding people are not happy with the fact that farmers are still largely using the 20, 30-year-old varieties. MTU 1010 is still the most popular around here. Swarna, Samba Masuri, you know, they're 25, 30-year-old varieties. And the farmers are not changing it because the customers want the same thing. Right. So I don't know which farmers you're talking about who.
And yeah, recently I visited Bihar, some area. And second question was, because in Bihar, this last 2 years, a more flooded area.
Right.
So farmers could not grow that rice. So is any policy like if that incident happened in some specific area, so government suggests some rice variety you grow because last 2 years facing you all these problems, so you can use this type of varieties, or but there is no any supports by the farmers. So any farmer or scientific team and targeted that area, or like it's
Yeah, so I can't say anything about the government policies, etc., but those kind of things do happen. And especially now, if we have all these apps that the farmers use for, you know, pests and pesticides and fertilizers, etc., one can build that into those apps that wherever farmers in the region are having problem, they can actually send the message. And there are organizations, IRRI does that also, to be able to take the relevant seeds. So flooding, for example, there is the submergence tolerance gene is there. And although that withstands flooding for 14 days, there are now lines coming up which can withstand flooding for 21 days, right? And yesterday, I was actually reviewing a project from Assam Agriculture University, and they are trying to put the flooding, the snorkel, and the aerobic germination genes all together in an elite variety. So those are the kind of lines which will become available easily. But yes, in terms of being able to immediately respond to farmers' needs, if the lines, the relevant lines and varieties are available, there are organizations, and farmers now can easily access those organizations through these apps, etc. So, yeah. Okay, good. Thank you again so much.